here. Green is online. And as a reminder, don't forget, you guys have to send the attendance words by 9 a.m. to be considered present if you are online. I mean, you do get the extra credit for the points, just like everyone else, I mean, the words, but it's more than just extra credit for you. It's also a part of your attendance. Harrison is here. Gibson is here. James is here. Kesterson is here. King is online. Don't forget to send the word before 9 a.m. Lewis. Yep, Lewis is here. Morrison is here. Grace is online. Don't forget to send the words before 9 a.m. Ward is here. Back to the top. Aki is here. That's that key back there, right? No? Sorry. Dawson? Here. Yep. Between it being dark and me not wearing my glasses and you sitting where he sometimes sits. Yeah. I confused me. Ellis is here though, right? Ellis? Right, with headphones? You probably can't hear me. That's him though, right? Yeah, good enough. Everybody else is accounted for. Yes. Who else is online? We've got King, James, Grace, and Green. King, James, Grace, and King. All right, everybody's accounted for. We've got 3 minutes. Pull up the PowerPoint. Someone messed with my PowerPoint the other time. People trying to sabotage me. Linda Q is here. He is here. Is anybody affected or has family affected by those the floods or the hurricane? I know Morrison, well, you said you live on the East Coast, right? So, yeah. I know they got a little bit of choppy weather, but that's about it. Anybody else? That's good news. Very good news. Anybody watch college football this weekend? I didn't get to see WVSU play, but the other game was pretty good. Except for my own team. Never seen a fall from grace as big as Florida State has done. It is what it is. Is that Paula back there? Yep. Paula is here. All right, let's do some unimportant announcements since we have less than a minute left, and then I'll do the more important ones once it's time. So we are starting Module 4 technically, which means in 2 weeks, that's when the next exam is due. Most likely we'll be done with that material because this is the last chapter for that exam. We'll be done with that material very early. Doesn't mean you have to get the exam done early, it just means you have more time to take it. Remember, the quizzes are the study guides. So take those quizzes as many times as you can. One of your classmates did the smart thing, and after they took the quizzes, they met me online so we could talk about the questions they missed. So they knew the right answer, and they knew why the wrong answer was the wrong answer. And they did very well on the last exam, so make sure you take advantage of that. Parks is here. Also, again, to remind you, you should really start working on independent work. 100 points worth of work, if you start from the beginning of the semester, it's not that hard. If you start now, It's going to be a challenge. If you wait till the end, you're not going to do it. And it's a full letter grade. Remember, you got 1,000 points for the whole semester. Independent work is worth 100 points. So that's a full letter grade, and it's completely in your control. Just start writing papers, or all these times I ask you questions, and I say, send me the answer. Send me the answer. I know that's only worth 1 point, but every little bit's going to help. I challenge you, at least since we are, again, starting module 4, which is the middle module, at least write one paper between now and the end of module 4. Because for those of you who have written papers and turned them in, and you could probably attest to this, it's not as easy as people think. I don't just read it like, oh, perfect, and give you your points. I'm digging into your sources. I'm making sure everything you wrote matches your sources. I'm making sure you didn't plagiarize. I'm making sure you didn't copy and paste it and just change a few words, like, use synonyms. So start sending your stuff, because it's not necessarily hard. Once you understand how I grade, then it's not hard. But if you just think you're going to turn a bunch of stuff in and get full credit, you might be wrong. Hopefully you're right. Hopefully you're really good at writing. But I'm here to challenge you. Anyway, any questions about anything? This week is a regular old week. You should have already had your pre-lab done on Friday. And then those extra other hoops that some of you got to jump through should be done. I took the weekend off, so if you haven't turned anything in that was due on Friday or Sunday, now's the time to do it. Because as soon as we're done here, I'm going to go to my office and start grading everything from the order in which it was received, right? So as long as you get it in before I'm done grading, as far as I'm concerned, I consider that all done. So any questions? Good. Let's jump back into photosynthesis. Where'd that go? Excuse me. Come on. Maybe if I X out and come back in. Don't know what happened there. So, to remind you where we left off is, we did the overview of photosynthesis, and then we started getting into more detail. We went back after we did the overview and talked about everything in more detail. And this is where we left off, was this little last explanation of the light reactions. Hold on, let me also make sure that, this is killing me. Technology's killing me today. Let me make sure we're still recording before I move forward. Okay. Good. All right. Anyway, this is where we left off, right? So we're basically done talking about the light reactions. So before we jump into the next stage, remember there's only 2 stages in photosynthesis. Let's do again a really quick overview of photosynthesis. And this, even though this picture is more of a metaphor than actually what happens, we could still talk about what happens. And this is the last thing we saw last week. Speaking of which, weak will be the first word for attendance. Weak, like not strong weak, but a weak part of the calendar. Anyway, so when you think of light reactions, you should be thinking, this is the energy, right? This is the stuff that's going to provide the energy that we need to make glucose. Well, first of all, when you think of photosynthesis, that's what you should think of. We're trying to make glucose, right? So in the process of making glucose, like, where do we get the energy to make the glucose? And that is what the light reactions are all about, right? Because what happens is water donates electrons, right, which produces oxygen in the process. Some energy energizes the electrons, and the primary electron acceptor captures that high energy electron and then basically brings it to this electron transport chain, like the same kind that we had in respiration. That high energy electron goes from protein to protein, which pumps hydrogen ions against their concentration gradient. Then they come back down, and then that process of coming down, we make a bunch of ATP, just like we do in respiration. But again, now this electron has lost some of its energy because it went through the electron transport chain. So we need to energize it again. So then again, sunlight energy, a photon, energizes it. It's even higher energy now. It gets captured once again. And this time it's incorporated into the NADPH, or in other words, the bucket that carries the electrons from here to the next stage that we're going to talk about, which is the light reaction. The Calvin cycle. So again, the whole purpose of photosynthesis is to create glucose. The whole purpose of the first stage of photosynthesis is to create the energy to make the glucose, specifically the ATP, because it's going to take energy to squish those carbons together. And then, of course, these high energy electrons because we're going to incorporate those high energy electrons into the glucose molecules. And hopefully by this point, you're just sick of hearing me say high energy electrons, because I've been saying it a lot. But yeah, so remember when we talked about respiration, that's what I kept talking about, right? We break down the glucose to capture the high energy electrons, right? Then we break down the pyruvate to capture the high energy electrons. It's all about those high energy electrons in respiration. So this is where they came from. So any questions about the light reactions?
Is this going on during the fall when the leaves change colors? Is this still going on?
To a much lesser extent. That's a great question. And you can look that up for independent work too, because to remind you of what we talked about last week, even though I told you guys didn't need to know it for the exam, what happens is, like right now, even though it's still fall, it's still mostly everything's green, everything's there. Chlorophyll A, chlorophyll B, all the carotenoids, which gives it its color. And then once things start getting cold and the days start getting shorter, then the chlorophyll leaves, and then we only see the carotenoids, the oranges and the browns and reds and stuff like yellows. So once the chlorophyll is gone, that's a good question, and I actually don't remember the answer. So, because remember, those other stuff, the carotenoids were there to protect the chlorophyll from getting too much damage from UV light. That's the purpose. But now that I'm thinking of it, either I never found out or I don't remember if they photosynthesize. So that's a great question for independent work. Anyway, that's a great question, a good question. Before I move on, another independent work idea, as always, everything I tell you, this is such an easy independent work topic. Look it up and confirm everything I'm telling you is true. Is it true that electrons, a photon excites an electron once and then it does it again? Yes, I'm telling you that. Yes, your textbook tells you that, but find the non-textbook source. But yep, I confirmed it. According to this source, this is true. Or if you really want to dig deeper, how do we know all this? Anything I teach you, right? Like, how do we know that a photon first excites an electron? How do we know that water is what donates the electrons? Who did those experiments and how did the experiment work? Where we figured those out. Anyway, moving forward. So again, a big overview of what we talked about before we move on to the next section. ATP production in the light reactions, which is what we're still talking about, that first stage, is similar to the ATP production and respiration, right? Because in both, we have these high energy electrons that are going to the electron transport chain, and they're going from protein to protein. And then when they're doing that, they're releasing their energy. And that energy is really used to pump hydrogen ions against their concentration gradient. Back to transport, remember? And then because they're against the concentration gradient, nature just takes its place and they diffuse back down the concentration gradient. And when that happens, we make ATP, right? Specifically, and it's not on here, but we've talked about it before, the enzyme ATP synthase makes ATP, both in photosynthesis and respiration. And of course, I'm going to show you a picture of this on the next slide. Here's some differences though. In respiration, it's the food that provides those high energy electrons, right? We focus on glucose, so for the exam, we're just going to say glucose, right? We're going to forget about the fact that we also eat, monosaccharides, disaccharides, polysaccharides, lipids, proteins, right? We eat all that stuff, but for the exam, we just focus on glucose. But yeah, in respiration, it's the food that's providing those high energy electrons. In photosynthesis, it's the water that provides the electrons, and then it's the process of photosynthesis that captures that sun energy to energize those electrons. And of course, that process, when the water produces the provides the electrons, that's where we get our oxygen from. Anyway, so any questions about this slide before I show you a visualization of photosynthesis? Of the light reactions of photosynthesis. All right, so there it is once again. Here's the whole chloroplast. We're zooming in to this one particular thylakoid, right? So we can see inside the membrane. Here's where this is all happening, photosystem 1, photosystem 2. So again, in your brain, try to picture that first picture I showed you this morning of them hitting the sledgehammer and the electron getting excited and then losing some of its excitement, but then getting even more excited. That's what's happening here, right? This is not a metaphor. This is what's actually happening. Water provides the electrons. Technically, there's 2 of them. You don't need to know that. In the process, you're providing, it makes oxygen. Those electrons get excited from light. Then they go from protein to protein in the electron transport chain, releasing their energy. That energy is used to take these hydrogen ions and pump them against the concentration gradient. These electrons get excited again because they lost some of their energy right here. They get captured by NADPH, or NADP plus, turning it into NADPH. Those high energy electrons are then taken off to the Calvin cycle. These hydrogen ions that were pumped against their concentration gradient can now diffuse back down the concentration gradient through the enzyme called ATP synthase. Of course, that process produces ATP. So then here we go. We have ATP and high energy electrons that go into the Calvin cycle. So again, I think if you can just remember that the first stage, light reactions, is all about providing the energy to the Calvin cycle, then hopefully everything else can make sense. A lot of the test questions should make sense to you. Like you might forget it, but hopefully you can just figure it out in the test, right? When I say which of the two stages uses light energy, well, first of all, light reactions, it's in the name, right? Or which of the two stages produces ATP? Hopefully you'll remember, all right, the first stage is providing energy to the second stage, so therefore the first stage would produce ATP. Which of the two stages produces high energy electrons? The first stage. Hopefully that's all, I don't want to say common sense, but it's intuitive, right? The first stage is all about gathering energy, because the second stage is the stage that actually takes the carbon out of the air, squishes them together to make glucose. So if you can remember that, hopefully every single question on the exam for the photosynthesis part should come easy to you. Oh. Anyway, any questions about this slide? Okay. Well, the last slide, this one we talked about earlier, this was kind of comparing and contrasting the light reactions to respiration. So there's the light reactions, here's respiration again. Again, very similar, right? We have high energy electrons going from protein to protein in the electron transport chain. It's giving off its energy. That energy is used to pump hydrogen ions against the concentration gradient. Then they just do what they naturally do, which is diffuse back down the concentration gradient. And in that process, the enzyme ATP synthase produces a bunch of ATP. And of course, the electrons have to go somewhere. In this case, they come to the oxygen, which forms water, right? So again, this part right here is the part that's opposite of photosynthesis, right? Because photosynthesis provides the electrons, and that's what produces the oxygen. Whew. Anyway, that's a review picture, obviously, because it's about respiration. We're done talking about respiration, but are there any questions about that? All right, which brings us to the last main bullet point of this chapter, which is the Calvin cycle, making sugar from carbon dioxide. And you know what? If I remember correctly, I think, not that this matters too much, but it might be worth pointing out, I think I kept this PowerPoint the same as I did from the previous textbook. So I think if you look at your textbook, obviously it's going to have the same information, but it might be in a slightly different order. But anyway, just want to throw that out there. The next word for attendance, I'm going to point at it with the arrow. There you go. Next word for attendance. And again, if you're online, you have to send those words before 9 a.m., otherwise you're considered absent. And I have to report attendance periodically to the university. All right, so let's talk about the next cycle, or the next step, which is the Calvin cycle. Before I even talk about this, first of all, I want to remind you of some things I said earlier when we were talking about respiration. I said when you think about carbon, you should think about a cycle, right? Because in respiration, it's a citric acid cycle that produces carbon dioxide. Here, it's the Calvin cycle that uses carbon dioxide, right? So no matter which one you're getting tested on, whether it be photosynthesis or respiration, you should associate carbon dioxide with the cycle. And I will tell you this, I'm also not going to try to trick you. So there will not be any question where I say, which of the two stages uses carbon dioxide, right? And then, well, that part wouldn't be tricky. But then what the part I wouldn't do that could be tricky is if in the multiple choices I said, was it the Calvin cycle or is it the citric acid cycle, right? I'm not going to be tricky like that. So any question that has to do with photosynthesis, think all the options are going to be photosynthesis options. And any question that's about respiration, I think all the questions, all the options are respiration options. I'm not positive on that. I'll double check when I get a chance and get back to you. Anyway, let's talk about the Calvin cycle. Again, big picture. The whole purpose of photosynthesis is to produce glucose. And so far we haven't talked about that, right? We haven't really talked about how that's done. So remember, the last thing we just talked about is all about producing the energy that's going to be used by this cycle to produce that sugar. So there we go. Let's talk about it. The Calvin cycle functions as the sugar factory in the chloroplast. So again, thing we just got finished talking about, the light reactions. You could think of this whole process. This whole thing is like a factory that produces glucose. In the last cycle, that's our solar panels, right? That's the thing that's providing the electricity, so to speak, to the factory. One thing I'm not going to ask you about, but I'm going to discuss it so you understand how it works. The Calvin cycle regenerates its starting material with each turn of the cycle. And that'll make a lot more sense when I show you the picture. And basically, that's just why it's a cycle, right? Because we start with something, we do some things to it, and then by the end of it, we're back where we started, right? That's just what makes it a cycle. Actually, I'm going to put an X through this as a visual indication that I won't ask any questions about that. Each turn of the Calvin cycle has chemical inputs and outputs, just like any cycle would. Just like, so far you've only learned about two other cycles, the citric acid cycle, right? So you knew the inputs from there, and you knew the outputs. We also talked about the ATP cycle, which the inputs were ADP and a phosphate, and the outputs were ATP, right? Easy cycle. And then now here we go, there's another cycle, and we are going to talk about the inputs and the outputs. And you will need to know the inputs and the outputs. So for example, you need to know that we need carbon dioxide, right? So which of the two stages uses carbon dioxide? Like I said earlier, Calvin cycle. Which of the two stages uses ATP? Not makes, but uses. It's the Calvin cycle. Which of the two stages uses high energy electrons that happen to be carried by NADPH? That is the Calvin cycle. Which of the two stages produces glucose? And I know it says G3P, but to keep things easy for the exam, the question is going to say glucose. So that's probably the first time you've seen, definitely the first time you've seen G3P in this class, I think. But probably, or maybe the first time you've seen it in your life. But yes, technically, photosynthesis makes something called G3P, and that G3P is then used to make glucose or other stuff. As far as we're concerned, to keep things simple for the exam, photosynthesis produces glucose. So again, the inputs of the Calvin cycle are carbon dioxide from the air, and then ATP and high energy electrons from the light reactions. And of course, the only output that we're concerned with is glucose. Which is another test question. Again, not test question, another independent work topic. Amongst the things I said earlier, which is like question everything, like, verify that the CO2 comes from the input for Calvin cycle is CO2, and that it uses ATP and right? Everything I'm telling you, find another source and verify it if you want. And then the other question is, how do we get from G3P to glucose? Right? If you look that up, there's an answer to that. What's the name of the enzyme or enzymes that do it? Anyway, any questions about this before I give you a visualization of the Calvin cycle? All right. This picture is not from your textbook. If I remember correctly, the one from your textbooks, I don't know, it's a little bit harder to follow. So take a look at the one in textbook, take a look at this one, and whichever one you find easier to study, I would recommend studying that one. All right, let's pay attention to this and see if anybody notices anything. Some basic math here. And I know none of you are chemists or biologists, but take a look here at this picture. Here, the starting material for the Calvin cycle is this RUBP sugar. 1, 2, 3, 4, 5. It's a 5-carbon sugar. Just happens to have 2 phosphates attached to it. That's important, but not for us. Right, so that's the starting material. And then it meets together with carbon dioxide from the air. And then somehow we're left with a 3-carbon molecule. So does anybody see anything weird about that picture right there? Just look at that, think about it, do some basic math. And what is weird about this? This is like one of the flaws from the old textbook. So I'm telling you, that 5-carbon sugar is attaching to another carbon, and then the result of that is a 3-carbon molecule. What is wrong with this picture? Think about basic math. All right, I'll make it even easier. Here's a hint. 5 plus 1 equals what? Good, 6. And how many is this? 3, right? So if you're a thinker, hopefully that would honestly piss you off. That would piss me off if I was reading that without any explanation. Like, how do we go from a 5-carbon molecule and we add another carbon, we're left with a 3-carbon molecule? Well, the reason for that is, as much as I like this picture more than the one that's in your textbook, there are some flaws because there's actually 2 of those 3-carbon molecules. So this is actually skipping some steps. This RUBP sugar is attached to another carbon from carbon dioxide. So then you're left with a 6-carbon molecule. And then that 6-carbon molecule is broken in half, and then that's where we get 2 of these 3-carbon molecules. Now for the exam, that's fine, because you don't even need to know that the starting material is RUBP sugar. I'm not going to ask you that. You don't even need to know that the starting material is a 5-carbon molecule. I'm not going to ask you any of that. You don't even need to know that we start with this 3-carbon molecule after it's attached to the 5. Right? You don't need to know any of this. So far, everything I've shown you is not necessary for the exam. But there are some great test questions. Excuse me, I keep saying that. There are some great independent work topics here. So what happens when we add another carbon to this RUBP sugar? It's going to be a different molecule. What is that molecule called? What enzyme makes that reaction happen? Then, what reaction is it that breaks that 6-carbon molecule down into this 3-carbon molecule? And what enzyme did that? And what is the name of this 3-carbon molecule? So many questions, so many things you don't need to know, but you could look up for independent work. Anyway, here's where it gets a little bit more interesting as far as the exam is concerned. Finally, now that we have this 3-carbon molecule, we use some ATP, right? We use some energy, we incorporate some high energy electrons, and we turn this 3-carbon molecule into a G3P sugar. Which is interesting again to me, because again, this is not very important for the exam, for this 100 level non-biology major class, but when you look at this, these things basically look the same. Here's a 3-carbon molecule with a phosphate. Here's another 3-carbon molecule with a phosphate. So for independent work, once again, you could look it up. Like, what's the difference? First of all, like I said earlier, what is the name of this 3-carbon molecule? Second of all, how is it different? How is G3P different than that? And also, again, what enzyme made this reaction happen? And also, are there other reactions in between this reaction and this reaction? Hint, there are. Anyway, there we go. We have the the G3P sugar, but once again, to remind you, we have this 5 plus the 1, so technically there's 2 of these 3-carbon molecules. Therefore, there is actually 2 of these G3P sugars. Next step in the cycle, 1 of those G3P sugars leaves the cycle and goes on to make glucose and other stuff. And again, that's another great independent work question. What other stuff are we talking about? What else does G3P make? Anyway, 1 of them goes on to do that. The other one goes back into the cycle. So that G3P sugar stays in the cycle. It goes through another chemical reaction. Whoops, hit the wrong button here. Goes through another chemical reaction. We add another ATP to it. And then obviously, again, look at this. Here we have a 3-carbon sugar with 1 phosphate. And now we add another phosphate. That's accounted for. Well, where do these other 2 carbons come from? Again, for you guys, it doesn't matter, but it is worth looking into for independent work if you want. So how do we get from G3P sugar to RUBP sugar? Where do those other 2 carbons come from? What enzymes were responsible for those chemical reactions to happen? So this is a very, very simplified version. If you want to blow your mind and if this ever feels overwhelming to you, just look up the real version of the Calvin cycle and you go, okay, this is indeed the Cliff Notes version. Anyway, any questions about the Calvin cycle? Okay, here's a picture, another picture of it. Just kind of a big picture. As a reminder, right, all of this is taking place in the chloroplast with light reactions, which we talked about last week, in which water donated electrons and light energy excited those electrons on the process that produced oxygen. Right, those excited electrons were used to make ATP and were captured, right? So those excited electrons are then incorporated into a molecule of glucose in the Calvin cycle. And it took energy from the ATP, from the light reactions, to bring those carbons together to make that stuff happen in the Calvin cycle. And then, of course, even though this won't be a test question, since we used ATP and we used high energy electrons, what's left after the Calvin cycle, obviously other than glucose, which is what we're after, is the byproducts, which is a DP, right, because it's missing a phosphate, plus a loose phosphate, then of course NADP plus, which is the high energy electron carrier, just without the electrons, right? So it's now ready to go back to the light reactions and get another set of electrons, and then take them back, and get another set of electrons, and then take them back. When you think about it, in a sense, this is sort of a cycle itself, right? From light reactions to Calvin cycle to light reactions. Of course, your book doesn't describe it that way, and there are reasons it's not technically a cycle, but as far as that picture is concerned, and as far as the way we think about it, you could consider that a cycle as well. But anyway, any questions? Oh, wait, sorry, once again, there's a lot of test questions here, so let's just say it. Which of the two stages uses light? Light reactions. Which of the two stages uses water? Light reactions. Which of the two stages produces oxygen? Light reactions. Which of the two stages produces ATP? Light reactions. Which of the two stages uses ATP? Light reactions. Calvin cycle. Which of the two stages produces high energy electrons? Light reactions. Which of the two stages uses high energy electrons? Calvin cycle. Which of the two stages uses carbon dioxide? Calvin cycle. And finally, which of the two stages produces glucose, which is the whole purpose of this thing? Calvin cycle. A lot of test questions on that one picture. And of course, the big question that you're going to be asked, that again I mentioned last week, and it's going to make me sad when someone misses it, which happens in the chloroplast? Respiration or photosynthesis? Photosynthesis happens in the chloroplast, right? Respiration happens in mitochondria. Any questions about that? All right. Yeah, this is a review, but this is all stuff we just got finished talking about all this, so there's not necessarily any reason to review it. So that's it for that. Yes, question.
On that picture, you said that the RUBP wasn't going to be on the exam.
Nope. RUBP will not be on the exam. Not only do you need to not know that there's a 5-carbon sugar that meets up with the carbon dioxide to start the cycle, but you don't need to. Yeah, you don't even need to know that, much less the name of it.
Does that have anything to do with visco?
Yes. And you don't need to know that either. So the only name of any molecule that's used in light reactions that you need to know is carbon dioxide, which we use a carbon from the carbon dioxide. You need to know ATP is used, which technically produces ADP, because once you take a phosphate away, it makes ADP. You need to know high energy electrons that are carried from NADPH, and you need to know glucose. That's it. So yeah, your book talks about a bunch of other stuff, but I don't get that deep with the exam. Any other questions? In that case, I'm going to pull up the next chapter. We are doing good time here. And while I'm pulling it up, does anybody have any questions?
Do you make the exam or is it automatically for
I made them years ago, and then every semester I change them up a little bit. So, yeah, they were made by me, but they're all slightly different every semester. Which is why, sometimes I question myself. As you notice, I'm like, did I have a question about that? Sometimes I hesitate to say there'll definitely be no question about that because I don't remember if I put it on there or not. Any other questions? That was a good one. So while I'm doing this would be a decent time to remind you. November 11th, the class will be online only. So we are having class, we will not meet here on November 11th. Because it's Veterans Day, and the school doesn't give off Veterans Day, but that's as close as I'm getting, so I'm doing it. But like I said last week, as far as I'm concerned, in some senses, you guys are my boss. So, if any of you has a problem with that and actually does want to meet here in class on Monday morning, November 11th, let me know, and I'll just cancel my plans. Come on. What's going on here? Oh my goodness. That was the problem. I was just rushing it. Sorry I didn't do this earlier. Like I was mentioning, for those of you who were here early, I came in with 10 minutes. That should have been enough time to do this, but instead I fought with technology a little bit. Make sure, okay. Still recording, that's always handy. Chapter 5 is what we just finished. So, chapter, where are you at? Chapter 6, reproduction at the cellular level. We download this PowerPoint. We'll get started slowly on this one. I would rather, never mind, let's just get started. PowerPoint, chapter 6. Yeah. So just to make sure you're very aware of this, what we are about to start, we're actually a little bit ahead of the game somehow. We were behind, now we're ahead. What we're about to start will not be on this next exam, right? So we just today, when we finished photosynthesis, we just finished the things that'll be on the second exam. Right? So for the second exam, it's mostly going to be about energy, right? Photosynthesis, respiration, kinetic energy versus potential energy. How do enzymes work? Activation energy, all that stuff is what you're going to be tested on for exam number 2. Here we go. This portion of the exam, so exam 3, excuse me, I should say it that way, this portion of the semester is going to be all about cellular reproduction in a sense. That's exactly what this chapter is about. But then the following chapters that you're going to be tested on are basically variations of this, right? So first we're going to learn about how cells reproduce. Then we're going to learn about genetics. And of course, genetics in a sense is basically the result of cellular respiration. So again, the exam you are about to take, or the exam for the module that we just finished, is all about energy, which is one of those properties of life. This one is all about cellular reproduction, right? Another property of life. So there we go. We're just kind of rolling through these properties of life. These pictures are from your old textbook, but we have plenty of time since we're a little bit ahead of the game. So let's play the old guessing game. What is cellular respiration? Why does this matter for you as regular day people? I don't remember what your textbook was getting at with some of these. But first of all, does anybody know what these are? Right here. Those are boogers. So the next word for attendance is boogers. That's only half true. Boogers is the next word for attendance, but those are not boogers. Those are chromosomes. And the old textbook, what they were getting at with that picture, of course, this is a bad analogy. It's a true statement, but this is verbatim from the old textbook. Keeping track of chromosomes during cell division is vital. Duplicating the wrong number of chromosomes is almost always fatal. Well, I guess that is true. So basically, you wouldn't be alive if you didn't have the right number of chromosomes, depending on the chromosome. Sometimes you can get extra and be okay, and we'll talk about that in this chapter. Sometimes it'll kill you before you're even born. But that's for humans, which is another thing to look up if you want later. Keep that in mind too. And I think last week I brought this up. Like, for example, some people think that seedless watermelons are dangerous. They're not dangerous. What happens is we've produced watermelons that have too many chromosomes. Because of that, they can't reproduce. So just like you can technically eat a mule, I don't know why you want to, but a mule wouldn't be any more dangerous than eating a horse or eating a donkey. But a mule is the cross of a donkey, and a horse, and they can't reproduce, right? It's a healthy thing, it just can't reproduce. Just like a liger. A liger is a cross of a tiger and a lion. Ligers can't reproduce. But they're healthy. It's not like they're some kind of abomination of nature. Anyway, so that's what the old textbook was getting at with this. Here's a harder one, because now you're really just guessing. But can anyone guess what this, obviously that's an x-ray or some sort of medical imaging. What do you think that might be a picture of? That's obviously a lung area, but one's glowing, and one side's glowing, and the other one's not. So what do you think that might be? If you had to guess.
Cancer?
Yeah, great guess. And that's exactly what it is. That is lung cancer. So in this chapter, this is a very important chapter, because in this chapter, you're going to understand the basics of cellular division, and that's basically what cancer is. To understand what cancer is, all types of cancer, you have to understand cellular division, because cancer is when cellular division goes wrong. So again, unfortunately, most of you If you haven't already, will be touched by cancer in your life, either you or somebody you love, which means you're probably going to be doing some research about cancer one day in your life. So luckily for you, you won't be starting from scratch because I'm making you the basics in this chapter. Now this picture, here's a molecule of DNA. Obviously, it's not this big compared, like, right, because that's supposedly the DNA from this woman. Obviously, it's not one molecule of DNA is not as wide as a woman. But any guesses what the old textbook may have been getting at with that picture? I mean, this is really just a guess, but anybody know? I'll just read it from the old textbook. Your old textbook points out that if stretched out, the DNA in any one of your cells would be taller than you. Which is interesting when you think about the trillions of cells that we're made up of. So that's another easy independent work question that you don't even need to write a paper about. You can just look up the answer, and send me an email and get 1 point. If you were to stretch all the DNA in your body together, line them up, how long would that be? It's a very long molecule if you stretch them end to end. Every single, all 46 chromosomes in each of your cells, and then each of your cells. It's a very big number. Anyway, let's jump into it. This part was also not in your textbook, but I left it. I'm not going to talk about it, but I left it on the PowerPoint so you can read about it if you want. And this has actually been in the news, not just this shark, but other stuff recently. There are some, how do I put this? Regular animals, like animals that you think of when you use the word animals, you would think about some of these animals, and some of them can reproduce, they can clone themselves, basically. It's something called parthenogenesis. How many of you have seen the first Jurassic Park? Do you remember that they were like, their safeguard was that all the dinosaurs are females, right? Like, oh, they can't reproduce, they're all females. And then one did reproduce, that was basically this. Because they, some animals can reproduce by themselves. Actually, a lot of the basic ones can, but these more advanced animals can do it too. So again, independent work topic. Yes, there's the shark, but there's other things that have done this too, if you want to look that up. What are some other famous examples of animals that have reproduced without a mate? It's always the females reproducing without the males, which is scary, right? The males, what if humans evolved to do that, and the males would just be completely useless? Anyway, read about that if you want. I think it's interesting. All right, so this is an interesting chapter because it's broken down into only two main bullet points. What cellular reproduction accomplishes, and then the cell cycle and mitosis. Don't let that fool you. Even though there's only two main bullet points, there's actually a lot of material in this chapter. Lucky for us, since we're a little bit ahead, I can kind of slow down and make sure we spend time with this. Reproduction. Obviously. Actually, before I say that, or some of you guys already saw it, but let me ask you this. How many of you have reproduced? All right, I only see one hand going up. But you're actually all wrong, except Bree. You all have reproduced, and you all are reproducing right now as you sit there. Because usually when people think of reproduction, excuse me, back up here. Usually when people think of reproduction, they probably think what you guys all thought, which is like how many of you have had kids. But that's not the only reproduction, right? That's only how humans reproduce to create another human. But there are other, which is this first bullet point, but there are other types of reproduction. And more often, that's the case, right? More often, reproduction involves the production of new cells, right? So you all started life as one cell, right? Your dad's sperm met with your mom's egg, and at that point, you were one cell. And you've grown since then. How many of you are still one cell? None of you, right? So the fact that you're here and not one cell means you have reproduced a lot. So cells undergo reproduction. We also call that cellular division. I'm going to put it next to this, not because it's not important, but because that's not going to be a test question. I'm not going to ask you, like, what's the definition of cell division? Just know that in this chapter, in this context, when we talk about cell division, we are talking about reproduction at the cellular level. In this process, two daughter cells are produced. And I'm only going to put it next to this, not because it's not going to be on the exam, but actually, you know what? I won't put it next to that for that reason. That will be on the exam, but we'll talk about it in greater detail later. This is kind of an introduction to the concept. But yeah, when a cell goes through reproduction, it produces two daughter cells. And these two daughter cells are genetically identical to each other and to the original parent cell. And again, we'll go into that in much more detail later. But that is definitely something you'll need to know for the exam. All that information there. Of course, there's another type of reproduction too, and we'll talk about that, in which case this will no longer be valid. But for now, that is valid. Any questions about that? Okay. Before cellular division happens, before this process happens, then the chromosomes also have to duplicate, right? And you need to know what chromosomes are. Those are the structures that contain DNA. And they have to duplicate. As a matter of fact, I'll put a little circle through this. I like the fact that we're talking about it like this, because when you think about it, when a cell is reproducing, when it's duplicating itself, everything has to be duplicated, right? Not just the chromosomes, but everything in there, right? All the organelles need to be duplicated, everything. If you're going to take a cell and cut it in half, which is what we're going to do, we have to duplicate everything. But for this exam, or this chapter, we don't focus on everything that happens. We focus on what the chromosomes do mostly. So keep that in mind. Most of this discussion that we're going to have is going to focus on the chromosomes. But just keep in the back of your mind that there are other things going on too that we don't even talk about, that you could look up for independent work. And we will mention some of it throughout this chapter, but for the most part, again, we're focused on the chromosomes. And just so you know, cellular division plays the role in the lives of organisms. So it replaces damaged or lost cells, right? So if you get cut and it heals, for example, that's cell division. When you grow, again, like I said, you guys are not one cell. You used to be one cell, but you're not anymore. That's growth, right? That's cell division. And then there's even reproduction, the way you guys thought I meant reproduction when I first asked it, right? When Bree was the only one who raised her hand. There's also cell division that's responsible for that as well. So the production of a new organism is also due to cellular reproduction. So any questions about this slide? It's all very introductory so far. All right, here's some examples. Obviously, this is just an example. I'm not going to test you on examples. Also, this picture is not from your textbook. This is from the old textbook. Here's a human kidney cell. You can see it's got like this line in between it, because actually, technically, this one human kidney cell is almost two cells, because that's how cellular division works. Here's an early human embryo. So again, you start off as one cell, and then that thing divides into 2, and then 4, and then 8, 16, 32, 64, so on and so forth. And that's what an embryo is, right? It just keeps doubling. Here's the reproduction of an amoeba. Again, it's just an example. You don't need to know about it, but you can see it kind of splitting in the middle. And for an amoeba, that's a single-celled organism. So when it splits its one cell, that's how it produces a new organism, right? So when we split new cells, that's how we grow and heal. But for a single-celled organism, that's just how they reproduce. Then there's things like this, which I also find very interesting. I don't know if you can tell what it is, but if you cut the arm off of the sea star, or starfish, however you want to say it, not only will that starfish grow a new arm back, but the arm that you just cut off will grow a new starfish. And technically, those two starfish will be clones of each other. That would be the same as like if I took you, cut off your arm, then your arm grew back, but then your arm also grew a new you. That would be, and then that other you would be a clone. So very interesting that it happens in the animal kingdom. That's another interesting independent work topic. If you read that, that would be more like a research paper, which you could do for independent work. Like, what is it about certain animals that allow them to do that? The more basic animals, right? Then you have more complicated, more evolved animals like us. We're supposed to be more complicated and more evolved, yet we can't do that, right? And then like there's intermediaries. So for example, for some lizards, if you cut their tail off, they'll grow a new tail, but their old tail won't grow a new lizard, right? So there's obviously some sort of like in between, right? Like this, again, that's one style of reproduction. Then you have us, which if you cut something off, it's gone. And then there's in between. Anyway, and then there's also that example. You can also do that with plants. How many of you have ever done that with plants? Like if you cut off, I don't know, cilantro, yes, if you put it in water, it'll grow roots, and then you can plant that and it'll reproduce, right? So again, that's just a clone. You can make your own clones. Anyway, any questions about this slide? All right, the next word for attendance will be, let's say, C. C is the next word for attendance. As in a body of water, not as in I see you. All right, so there's two different types of reproduction. This slide is a great hint at that. Right? We have asexual reproduction and sexual reproduction. So let's talk about the difference, because there will be a few questions about that. You will need to know the difference between the two for the exam. For asexual reproduction, there is no egg and sperm, which should make sense, right? When you think of sex, you should think of egg and sperm. Therefore, if you think of the opposite of sex, which is asexual, you should think of the opposite of sperm and egg, which is no sperm and egg. So in this case, the parent, I really should put that in parentheses, because that almost indicates it's like a full-blown organism. And sometimes it is, but not always. But the parent and the offspring have identical genes. And I've already said that in the previous slide. Now I'm just saying, I just said it in different words. Right? So in asexual reproduction, when a cell duplicates itself, those offspring, if you want to call them those new cells, are genetically identical to the parent and each other. Of course, there are some exceptions that we'll talk about at the end of this chapter. Obviously, things go wrong. You have mutations. Sometimes chromosomes don't separate the way they should, and we'll talk about all that. So that is what single-celled organisms use for reproduction. So again, that picture I just showed you earlier, that amoeba, that's how it reproduces, right? So we don't use asexual reproduction to create new humans. We do use asexual reproduction as for growth and healing, but we don't create new humans with asexual reproduction. Well, not first. But some all single-celled organisms, that is how they create new organisms. Yeast, that's another single-celled organism, and it's eukaryote. It goes through asexual reproduction to produce new organisms. Also, like I mentioned earlier, and again, a lot of this is repetitive. It's just saying the same thing in different words. Many multicellular organisms, such as ourselves, use asexual reproduction. For us, we use it for, again, healing and growing. Again, using the examples I used earlier, sea stars can use it to grow new individuals, new organisms. Plants can use it to make new individuals. I will put it next to that, though. These are just examples. I'm not going to ask you about sea stars or particular plants. The point is that all single-celled organisms use asexual reproduction to produce new organisms, and sometimes some multicellular organisms can use asexual reproduction to produce new organisms. And of course, we use it in a way that's not meant for creating new organisms, but just for growth and maintenance. Any questions about that? All right, one more thing that you need to know about asexual reproduction. This one's important. What is the name of this reproduction? What type of cellular reproduction is it? And it's called mitosis. So when you think of mitosis, you should think of asexual reproduction. That is the cellular division that is responsible for asexual reproduction, which is mitosis. And again, just like the previous slide said, mitosis is what single-celled organisms use to create new organisms. Mitosis is what sometimes some multicellular organisms use to produce new organisms. Again, the plant or the sea star being an example. Or more often than not, the second bullet point right here, it is used for growth and maintenance of multicellular organisms. So again, as you're sitting here right now, you are going through mitosis, which is another decent independent work topic, right? What actually you'll kind of you'll get the answer to this when you do the lab, next week's lab. But you could ask yourself, what percentage of your cells at any given moment are going through mitosis, right? You could look that up if you want. Again, that would be a nice, easy one where you can just email me the answer for 1 point instead of writing a paper about it. What percentage of your cells at any given moment are going through mitosis? Anyway, any questions about this slide? All right, that's mitosis. That's for asexual reproduction. The other one is meiosis, and that's we'll get back to that when we get here on Wednesday. So that's it. You guys have a good week. If you haven't turned anything in from last week that you should have, do it now. When we leave here, I'm going to go start grading. And the good news for you is I have a ton of stuff to grade. So if you turn in something at 10 a.m., I probably still won't even grade it until this afternoon. Hopefully not that long, but the point is you have time. You have hope. I'll just say that.
2, 1.